Um, okay, it's my great pleasure to introduce Professor Lin Chao from UC San Diego. I would like to thank him again for accepting our seminar invitation. A brief background about Dr. Chao. Dr. Chao received his PhD from the University of Massachusetts, Amherst, and he was an NIH postdoctoral fellow at Princeton University. Dr. Chao joined UC San Diego in 1999, and he served as the chair of the section of Ecology, Behavior, and Evolution from 2004 to 2008. In 2014, he received the UC San Diego Chancellor's Associates Award for Excellence in Undergraduate Teaching. Dr. Chao is one of the true pioneers in the field of microbial evolution and ecology. And he is one of the unique scientists who uses both experimental and computational tools and theory to investigate fundamental processes in nature, such as ecology of host-parasite interactions, evolution of sex, mutations, ecology of transposable elements, group and individual adaptations, and lately, aging. He is one of the leaders in the field who made phages and bacteria model organisms to study molecular evolution. And I am personally a fan of how he elegantly simplifies complex concepts and makes difficult problems more approachable. Dr. Chao is the first PhD student of Professor Bruce Levin, who trained several very well-known scientists such as Carl Bergstrom, Richard Lansky, and Mark Lipsitch, who became public figures during the COVID pandemic. Dr. Chao continued this great tradition himself, and he is also known for his reputation for being a great mentor and a teacher. He trained several well-known scientists in our field, including Paul Turner, Martin Eckermann, Olivier Tenalion, and Dan Weinreich. Dr. Chao's seminar title is Stochasticity, Immortality, and Mortality in E. coli. And a couple of technical reminders. Thanks to Twitter, today we have audience from different universities. And another thing is today's seminar is being recorded, so please be aware of that. We will hopefully post it online if Dr. Chao approves. And please save your questions until the end of the seminar and make sure that your microphones are off. And please type your questions in the chat window, and I'll make sure that I will ask those questions to Dr. Chao at the end of the talk. Without further ado, I would like to leave the microphone to Dr. Chao. Thank you so much.
Thank you, Erdal. I'd like to thank for the invitation to present this seminar. And those of you who are here attending it live, I also thank you for being here. I come from a variety of backgrounds. I sometimes give talks to a more ecological and evolutionary audience. Sometimes I give talks to QBio. And today I understand this is bioinformatics, although I'm not primarily going to present the bioinformatic point of view. So if there's any kind of affiliation, it'll be probably more in a QBio approach. But because I often give different approaches and presentations, I kind of took the liberty to organize this talk by kind of jumping in directly without much of an introduction and giving you some details about a quantitative and computational study of aging in bacteria, and left the more general summary and approaches for the end. So let me first thank the people involved. This is the lab crew, past and present. Developing the project that I'll present to you has come through a very large effort, partly because we had to learn microscopy, and that was not something our lab had done. So these are the members who have helped us develop that. The early group is over here. Master students who help us collect the original data. Martin Ackerman, who Erdal mentioned, was my postdoc, and he planted into my brain the idea that bacteria could age, and a lot of the ideas that follow that I'll talk about. The remaining 6 people over here are current members of the lab that have worked at it. We've had the luck of having actually several very good students from Brazil through a program called CAPES that has sent Brazilian students to the United States. Camila is one who runs the lab and has actually mastered the art of microscopy. Audrey was a graduate student. Chao was a graduate student. Master's student Andrew. Mari was a summer intern. And Kristen was a lab assistant. With this group, we finally then managed to collect preliminary data that ended up providing us funding through NSF. Faculty at UCSD have also been very helpful, and some of you may recognize names here. Scott Rifkin, Terry Waugh, Jeff Hasty, Alex Gorsman, Sukjun Jun, brought to UCSD the QBio program, and more importantly from the perspective of this work, the microfluidic technologies for microscopy. So jumping into the specific problem, I'm just going to start off by giving you some details. Bacteria and other organisms can accumulate non-genetic damage, for example, oxidative in their cells. When I say non-genetic, I mean not damage to DNA, because that would be mutations. And for today's discussion, although relevant, mutations will not be the issue of discussion. So these are macromolecules that can become oxidized, proteins that can become unfolded, all sorts of effects that affect the function of a cell. Many of these damaged macromolecules will form aggregates. And the idea that I want to start discussing then is that when a bacterial cell, in this case E. coli, when a mother cell divides, a decision that it has to make is whether to distribute the damage that's in the mother asymmetrically or symmetrically. Now, of course, if the mother can repair the damage, it pays to repair it, but there may be costs associated with repair. So if the cost is too high, that damage will not be repaired, and that puts this dilemma into the mother's hand. Should she give the damage symmetrically or asymmetrically to the two daughters that she'll create by fission? And the answer is that she chooses to be asymmetrical. A lot of this comes from microscopy data, and I show you three of the approaches that we have used. The classical approach is to use agar pads, which is just a droplet of agar. The cells are put on the surface, and this is sandwiched between the agar pad and the cover slip, and that's put into a depression slide. That's been the standard way to do lineage tracking, growth of bacterial cells in microscopy. But recently, other developments have added better, more refined technology, and these are the microfluidics, which are devices which have in them very, very small channels on the magnitude of a bacterial cell. So here's one where there's a channel that's shaped like a cylinder or a tunnel with a blunt end. Bacterial cells are put in there, and with media flowing through the top, the cells then are fed, they reproduce, they replicate, and as they do that, they'll push cells, the daughters, out, and the media will wash them away. This approach is really good for quantifying the bottom cell. You can get very, very long-term equilibrium information on the cells, equilibrium respect to those culture conditions. Unfortunately, the daughters get washed away, so you don't have a chance to collect detailed information on the daughters. This other device we just call the large chamber is basically a chamber with a very low ceiling, so the cells are trapped as a monolayer. As a result, the residency time of cells in this chamber is longer, so that we can track some of the daughters for a longer time. However, there's also a limit, because media which flows along both sides where these arrows are will eventually wash the daughters as they are pushed out and removing them from the system, much like a mother machine. But you can get a few more generations, and we'll use that whenever possible. So these are the techniques that we will use, and I'll try to highlight them as we go to see how one was used instead of the other. I talked about aggregates. So this is a time-lapse microscopy of E. coli cells dividing. In this case, a protein, IDPA, which is a chaperone associated with the building of the aggregates of damaged proteins and bacteria, is labeled by fusing a GFP reporter. As a result, we can track the aggregates. And in this case, in this monolayer of cells, the main message here being that these aggregates tend to congregate to the poles, the ends of the cells. This is better shown if we follow it in the mother machine. So this is now narrow channels where cells are trapped at the bottom. You can see that the cell at the bottom tends to have the aggregate at the farther end pole, the pole facing downwards. These three have it. This cell down here happens to be a cell that did not have a large aggregate, therefore was not reporting it at the time the experiment was started. But if you follow this, you'll see the buildup. Right here, you start seeing a buildup of an aggregate, and eventually becomes as large as the aggregates in the other cells. Notice that as this cell grows in length and divides, the daughter above it doesn't have the aggregate. So this cell at the bottom, which we refer to as the mother cell, is keeping the aggregate of damaged proteins to herself, and the daughter is giving a new start, but without any of the damaged proteins, or if there's some small amount, small enough to be reported by this technique. This whole setup then allows us to give names. So let me just go through some words that I'll be using to describe the outcomes. So here we have the mother machine with the microfluidic device. A cell is trapped down here. Because E. coli is a rod and it grows by elongating and then eventually dividing in the middle, the poles that are formed at the middle, at the midline for the two daughters, this pole here and this pole here, are newly synthesized. Because of the trap and the mother cell is physically held in this position or orientation, and the fact that division happens above her, the pole up here will always be what we call the new pole, because it was produced when this mother cell was born and its mother divided right here. Therefore, the pole pointing downwards is referred to as the old pole because it was synthesized 2 generations back or longer. With this naming, we can therefore polarize any cell and realize that one pole will always be the new pole because it was formed the previous generation when the cell was born. If that's the case, as we follow this cell forward in time, the daughters they produce will also be asymmetrically named because the upper daughter gets the new pole of the mother, so we call it the new daughter, and then the bottom cell gets the old pole of the mother, and we call that the old daughter. Mother, 2 daughters, new pole, new daughter, old daughter. Notice that the aggregate, labeled in this case with the fluorescence, always remains at the old pole of the old daughter. Therefore, damage is allocated asymmetrically to old daughter. Damage accumulates in the old pole. As damage accumulates, the growth rate of a cell decreases and the cell ages. So this is saying that the old daughter, as it accumulates damage, can be shown to elongate more slowly, while the new daughter, without the damage or less of it, will be able to elongate more quickly. This is just a schematic of the same discussion I just had from the previous slide. Mother cell, old and new daughter. But what I've added here now are the ages of the poles. We start with the mother, realizing that one pole will always be age 0 because it was just produced at the last division. This is the new pole. The other pole is the old pole, and I here arbitrarily give it an age of 1. It's arbitrary because unless you follow a lineage, if someone gives you a cell, you do not know that age. I give it 1, but it could be millions of years going back to the first bacteria. But if we follow 1, as this cell elongates and divides, the new daughter will get the new pole. So this pole now has an age 1, and the new daughter has a age pole of 0 generations. The old daughter will get the one that was one generation old, so it becomes a 2. And we can go forward in time. This 2 becomes a 3. 1, 0 becomes a 2. New poles, that 0 becomes this 1. This is important for later discussion because as we track lineages in this way, we'll give them notations and basically realizing that this is the old pole, this is the old daughter that got the old pole, this is now the second generation that also got the old pole, so we'll refer to this daughter as the old daughter, and she is called OO because she got the old pole 2 times in a row. This one's called new because it got the mother's new pole. This was the mother of this cell. It was a new daughter, therefore new daughter, or new. So from the broad perspective, this will lead us to discuss and think about aging in bacteria because as the old daughter accumulates damage, it will slow down. I would like to argue that there are many analogies between aging in bacteria and multicellular organisms, and that's a topic that I'll pick up in the second half of the presentation. This also leads us to discuss then what's the evolutionary advantage of partitioning damage asymmetrically. The more direct approach, however, will begin by focusing on bacteria, aging, develop a computational model, and then use that to predict some experiments, and then I'll provide the data that we collected, attempting to test those predictions and then interpret them. So first, the model. The model is a simple rate model where we begin by describing the amount of damage inside a bacterial cell. K refers to damage, so KT is the amount of damage at any point time T. K0 is the amount of damage the cell was born with. And then lambda T is the rate at which the cell accumulates new damage. So what I have equals how much baggage my parents gave me and how much the environment hurts me with time. We call this the equation of life. Lambda is a rate constant, and simply as a function of time, I'll get new damage. We then assume that the bacterial cell has to do something in order to divide. And in this case, it has to build up a product that we call capital P. This product could be length. So in that case, bacteria will simply grow a certain length and then divide. Since we don't know exactly what is that's being counted in bacteria, I generalize the model to represent some product, capital P. The rate at which this product is built through synthesis is a function of the amount of damage I have. So I simply scaled it to be 1 minus the amount of damage I have at any point in time T. Substituting this equation into here, I can rewrite delta P. I can then integrate and solve for the amount of product I have at any point in time T. It is just a quadratic, which is equal to 1 minus the amount of damage I have times time, and then a constant that's a function of the rate of new damage, and then time squared. When the amount of product equals some threshold amount that we call pi, the cell will divide, So time becomes capital T, which is the doubling time of the cell. However much time the cell will take before it divides and makes two daughters, doubling time. Because I now have capital T or doubling time, I can put it back into the equation 1, and that allows me then to solve for some of the constants that allow me to get a solution for the model. And the solution basically gives me the amount of damage a cell has, a mother cell has at the time of division. K is the amount of damage, T is the doubling time, so this is the amount of damage the cell has at that point right before division. That damage, as I said now, will be partitioned to the two daughters, and it could be asymmetrical. So we have a coefficient, little a, which describes the degree of asymmetry. If little a is 1/2, 1 minus little a is also 1/2, and the partitioning is symmetrical. If, however, a is less than 1/2, anywhere from 0 to 1/2, then the partition will be asymmetrical. Once I have this, I know how much damage the daughters are born with, and I can just cycle through the equation again, the previous equation, because if I know the amount of damage a cell is born with, I can go through this iteration and give the doubling time of the daughters. So give me the doubling time of a mother cell. The model allows me to predict the doubling time of the two daughters. I will use the notation T1 for the new daughter, T2 for the old daughter, and T0 is the doubling time of the mother. The visualization of the outcome of this model is best done graphically. So in this case, it's a phase plot where the mother's doubling time, capital T0, is on the x-axis. On the y-axis is the doubling time of the two daughters, capital T1 and capital T2. Old daughter will always be red, new daughter will always be blue. Old daughter, remember, is the one that gets more damage. The mapping of the relationship between the mother's doubling time and the daughter's doubling time is predicted by the model to basically two quasi-linear relationships with a slope less than 1. The dotted line is a line with a slope 1, which is basically the identity line. In other words, when a mother maps to a daughter of the same value. So if the mother had a value of 25, then the daughter also has a doubling time of 25. That would fall on the dotted line. These are the two relationships that come out of the model. First question is there any validity to this? We'll interpret the significance in a second. To see whether this had any semblance of reality, we first then collected data. We basically follow through microscopy, get the doubling time of the mother, get the doubling time of the old and new daughter, and then simply plot it as such a graph. And what we see is a relationship that comes very closely to what the model predicted. Red dots are old daughters, blue dots are new daughters coming from the same mother. So every mother on this axis gives me two daughters. What we first did then was take the model and made a fit to the data. Basically, it's a fit on minimizing the residuals, and these are the lines that produce from the model for a fit that minimizes the deviation from the data. If you remember, the model had 3 parameters that I played with. One was pi, which is the amount of product it needs to divide. The other is lambda, the rate of new accumulation of damage. And the third one was the asymmetry coefficient. I show you here primarily the asymmetry coefficient, which we estimated from the data to range from 0.35 to 0.45. I'll give you more about this later, but I just wanted to use this to indicate that the data has some ability to predict at least the general properties of the model. What this indicates, therefore, is that the notion of asymmetry is being realized in the laboratory system with E. coli, and also appreciate that this is indicating that the new daughter is getting somewhere on the order of 30 to 40% of the damage, whereas the old daughter is getting somewhere between 70 to 60% of the damage. Appreciate that if this value of A was 1/2, in other words, the process was symmetrical, the red and blue dots should converge into one cloud, and the red and blue line should collapse into one line. The fact that we see two lines and the separation of the old and new daughters as red and blue dots indicates that there is a asymmetry in the process. These lines, given the fact that they have a slope that's less than 1, intercept the identity line. And this intercept becomes important because they represent stable equilibrium points. There's an equilibrium point for the new daughter, there's an equilibrium for the new old daughter. And they're stable because of the following perspective. Imagine that I had a mother that had a doubling time of 30 minutes. Her old daughter would be predicted to fall on the red line. So a mother with a doubling time of 30 minutes would have an old daughter that has a doubling time of somewhere around here, say 27, 28 minutes. This is the old daughter. But that old daughter in turn will become a mother. So when it divides, we'll project that number, say 27, 28, to over here, which is the mother line. That mother would have a daughter then would fall right here, which is about 26, 27 minutes. If we project then that daughter to become a mother, will be over here, and the next daughter will be here. So we follow a mother and the lineage, the daughters, granddaughters, great-granddaughters would all progress downwards until the equilibrium point. And the reason for that is because a mother with 30 minutes has so much damage that it is growing slowly. But when a mother grows in length to make a new daughter, that growth dilutes the damage because the new material made by the mother is damage-free from the perspective that it is very new and hasn't had a chance to accumulate damage. So that dilution is rejuvenating the daughter, and as a result, the daughter and its lineage will have faster and faster doubling time because they are being born with less damage. Well, those daughters keep on experiencing decrease in doubling time and further rejuvenation, meaning that they will go all the way down here. No, and that's shown by the fact that we have a mother that say down here with 24 minutes, her daughter will be, 21 minutes, her daughter would be 24, which means it grows slower because it is above the dashed line. So a healthy mother with very little damage will have a daughter, an old daughter, that has more damage. And that's because this mother was born with so little damage that she happens to be healthy. Imagine if she has 0. Well, she cannot remain 0 because there's a spontaneous rate of new damage. So as a result, the daughters must be worse off. They'll have a longer doubling time, and then the granddaughters will be here, the great-granddaughters here, and they'll again converge to this point and not go beyond, because if they did, they would come back. Therefore, this is a stable equilibrium point. If that is true, the new daughters will also have one, but the equilibrium are different. The equilibrium for the old daughter is higher because they have more damage. As a result, they take longer to divide. New daughters have less damage, so they divide in shorter time, and therefore these 2 points are displaced from each other. Can we verify this with data? We can. The way to do this is to basically collect information on this bottom cell in this mother machine and follow its elongation rate, its growth rate, follow its doubling time. This cell, as I argued earlier, because division is always above it, the new pole will point upwards and the old pole will point downwards. What that means then is that this bottom cell is always the old daughter. Therefore, information from this cell gives us lineages that should converge to this equilibrium point. And here's a tracking of an old daughter lineage over 70 generations. And basically, we expected it to cycle around this point. There's a lot of noise, variation, and stochasticity. And if we plot the centroid, it is over here. So the data doesn't fit the prediction of the model perfectly in the sense that we would have thought that the equilibrium was right here. But nevertheless, there is something that's attracting and keeping the cells at this region, at least not perfectly, but away from the new daughter equilibrium. How does the new daughter equilibrium fare? Can we collect similar data on lineages to see whether those lineages of the blue new daughters would converge to this point? Unfortunately, we cannot get the data from the mother machine because, as I pointed out, the new daughters are the ones up here and they're washed out. They're pushed out and washed away. So we had to rely then on using the larger chamber because we had a larger monolayer bacteria where we could follow old daughters for longer time. I'm sorry, new daughters for longer time. And this is the data that comes out of it. The data is not as rich as before, partly because we still are limited by the washout of cells, but you can see lineages of old and new daughters in the large chamber will aggregate and congregate on 2 regions. This one corresponds to old daughters around the old daughter equilibrium, this one corresponding to the new daughter equilibrium. This is a similar presentation where now x-axis is over time. So this is the same data as here, but stretched out over time. The new daughter is coming to an equilibrium that's slightly higher than the equilibrium for the old daughters in blue. The difference is small, right? Over here, we're talking about 19 minutes. Over here, not even quite 21 minutes. But the nice thing about microfluidics is that we can collect very, very large sample sizes, which now range from hundreds to thousands, and we can get the statistical power to show that these are significantly different. So a question that arises at this point then, okay, we have equilibrium, are they truly stable? One argument for stability is this graph that I showed you earlier. And the stability results simply from the fact that the slope of this line at this equilibrium point, the local slope, is less than 1. If the red line had a slope of 1, it will match the identity line. In that case, you have a random walk process because the mother would map over to the dotted line, and if there's any stochasticity, I would deviate from the mapping point, but then the daughters then will reside at that point as they come and flip down to be a mother to reproduce. In order to have stability, the slope must be less than 1. That requirement is satisfying because if I plot you all the data and then look at the best fit, either for linear regression or on the model, the slope at the intercept is 0.347. This is less than 1, so the first requirement is satisfied. The second requirement, however, for stability of this equilibrium is that the stochasticity of variance around this point is not excessively large. And the argument is that if it's excessively large, by chance, I could get fluctuations in this noise pattern so that I could end up being off scale. So, you know, here we're limited to 28 minutes, but if by chance the noise was high, I could go to 28 minutes and the next generation by chance go up to 50 minutes and then to 100 and then maybe even higher. And eventually that cell may not divide because the waiting time for this population, things are changing more quickly. So a cell that doesn't divide for hundreds of minutes is either washed out or removed from the system by other factors. But because we had this long-term data for 70 generations, we could estimate the variance in the mapping from mother to daughter, and that gave a standard deviation of 0.07. So the combined requirement for stability is that the square of the slope, m, right here, and the standard deviation add up to something less than 1. If we do that, it is less than 1, 0.125. So this system and these equilibria are indeed stable. And we can kind of show you this with a simulation. So these are simulation data of populations where we kept everything at the parameters that we estimated, but now we added a noise term that's over here. So you can see when sigma 1, sigma 2 is 1.0, 1.5, this is excessive noise, and we start having deviations here in the gray where the doubling time is going over 1 million, right? So these are conditions where the cells could divide if we gave it 1 million minutes, but in reality, they're as good as dead. The green line is what happens when we have a noise level equivalent to what we observed. Sigma 1 equals 0.07. We magnify it for you here. So these are repeated simulation traces with the observed noise level, 0.07. And this is just real data to compare. They're all obviously going to match well because we're estimating the parameters. But this is just to illustrate the consequence of changing only the noise, nothing else. The stability through the slope is still there, but the noise level stochasticity is too high, and we can have losses of cells by chance alone. If these are stable equilibrium points, a question we can ask then is, where will the population of cells reside? Will they reside only at these points, U daughters here, O daughters there? Or will the population show other doubling times? The answer is the latter, and that's because if I have a old mother, which is the old daughter of its mother, it will be residing over here at 26 minutes. When that equilibrium mother divides, she'll reside right here at 26 minutes. Her old daughter will fall right here because the old daughters and the old granddaughters and the old great-granddaughters are a lineage at equilibrium. But the new daughter produced by this equilibrium mother will fall in the blue line right here, which is about 24 minutes, which is not at the equilibrium value at 23. So this new daughter must, through lineage replication, march down to this equilibrium. As a result, what happens in a population is a series of lineages that can reside here, the old daughter will, but the new daughter will pop up over here and have to come down here. And the population then is more like a series of networks where lineages will connect to new daughters, back to new daughter equilibrium, produce an old daughter that will then, through lineage replication, get back to the old daughter equilibrium. Realizing that also as this happens, there'll be old daughters that will pop upwards. The whole population then will consist of cells at equilibrium and a network of lineages that are moving back and forth. We can show that this happens in our studies because if we track lineages that are at the new daughter equilibrium, its old daughter will form a lineage that will go back to the equilibrium. And likewise, cells that come from the old daughter equilibrium will track down to the other equilibrium. So this is aging, this is rejuvenation. The outcome of this is that the cells then are immortal. Cells never go beyond and accumulate so much damage that they die, because once they reach this point, they achieve an equilibrium density of damage, and they are therefore immortal. Another point that I actually indicated earlier, appreciate that if we have symmetry, then these two red lines collapse to one line, and I have one equilibrium point. With one equilibrium point, there's no aging. Whenever I argue that this is a form aging, the first response that I often get then is, how can this be aging if there's no mortality? Isn't a characteristic and hallmark of aging that organisms die? Is mortality possible in this system? If we play with the model, we can find that mortality is possible, but to do that, we have to increase the rate of damage. Remember that one of the parameters in the model was lambda, and that was the rate of spontaneous damage. If we elevate the rate of spontaneous damage, the outcome of that is the old daughters, the ones that get more damage, will eventually be unable to achieve an equilibrium, because the new damage that accumulates every generation forces them to divide longer, longer, and longer, and the line never intercepts the identity line, so their doubling time will simply grow until it's infinitely long. In this case then, lineages are no longer immortal. Mortality will have been brought in simply by the fact that the rate of damage is higher. The species or the population will not go extinct, because populations are still able to survive because the new daughter equilibrium is able to persist. The population persists because this equilibrium is still possible. However, once a new daughter is produced, it will produce then old daughters, and then the old daughter lineage will die out. In that case, mortality is brought in. We can test this by increasing the rate of damage. And in this case, I've used the amount of light in the microscope, phototoxicity, as an agent of damage. So in this case, we're giving it basically FTSE, the same light that you use to excite GFP, and different levels, 300 milliseconds. So this is 3 seconds of light. Doubling time of the equilibrium, which are the points that I've been showing you earlier, are down at 20 to 25 minutes. But as I increase the damage rate through light, the old daughter equilibrium increases at a faster rate than the new daughter, and eventually above a threshold of 1,500 milliseconds of FTSE light, the doubling time increases to be longer than 200 minutes. This then is basically the effect of damage and showing that mortality now is introduced. This is another graph showing the trace of the loss of stability. So I showed you this earlier. This is now the trace with high levels of phototoxicity. The variance is higher, and eventually one cell basically accumulates to the point of having a very, very large doubling time. This is then a reanalysis of the stability. But what we do now is the same stability that I showed you earlier. We're now showing the slope M, stochasticity through sigma, as a function of the amount of damage through phototoxicity. As we increase the amount of phototoxicity, the slope goes from earlier values of 0.3 to very, very close to 1. So it's borderline unstable as the amount of damage is introduced. Sigma also increases with damage, and in this case, up to a value of 0.3. But if we do then the final analysis, which is the squared sums, if you notice here, the old daughter now is crossing the threshold of 1, which is why the old daughter lineages are going to extinction, because their doubling times are increasing to be very, very long. The new daughter is still below a threshold of 1, although there are ways of fluctuations that could bring it up high, and those windows could lead to extinction of the new daughter lineage. But on the average, the mean value is still less than 1, therefore allowing generally the population to persist. We get the same effect if we use streptomycin at very low doses, 5 microgram levels, which are sublethal concentrations. But streptomycin causes misfolded proteins that exposes them to oxidation. Misfolded proteins are also less functional, therefore damage. All this gives us another mechanism to increase the rate of damage, and the same pattern is observed, although in this case, streptomycin, by increasing the slope, is sufficient to make both equilibrium points unstable. However, notice that it is always the old daughter that experiences higher instability, whether through the slope or through the variance sigma or the final combined sums of squares. These are some final details about the model. This is simply showing the amount of FTSE, in this case, in seconds that we gave the system. And this is the damage rate that we estimate through the model, okay? I give you this only to indicate the model is able to re-extract the linear relationship between damage through phototoxicity that we introduced, and gives it back to us as an estimation of the parameter lambda in a linear fashion. So that doesn't mean that the model is 100% correct, but it indicates that the model is behaving in a consistent way as we would expect if it was reflecting the linear input of damage and the linear consequences of the effect. This is the asymmetry A that we estimate. And if you notice over here is lambda now, the amount of damage that we introduced, and the asymmetry value. The range 0.35 all the way up to 0.48. I gave you that range earlier in the slide. This is an interesting result that we don't quite understand. For some reason, as the rate of damage increases, the asymmetry actually decreases to be almost 0.5. We had anticipated the opposite. We thought that bacteria would be smart. More damage, it pays to be more asymmetrical, because if the old daughter is going to be dead with damage, why not give it everything? This way, the new daughter is born rejuvenated and as free as possible. So we expected this to go down, but it went up. We have to face this as the outcome. The best interpretation we can give is that this will indicate that whatever is a process that allows bacteria to be asymmetrical, it is saturating, somehow being overwhelmed as the damage rate goes up. Or another way of saying, as damage rate goes up, everything ages, including the system that created the asymmetry. So this is a summary. Given that I'm running low on time, I'm not going to recapitulate it. And just finish by giving out some final thoughts on how this may relate bacterial aging to aging in organisms, eukaryotes, and multicellular organisms in general. I've been presenting to you this view of bacteria. A mother produces an old and new daughter, red and blue. This daughter then produces its daughters, which are again old and new and red and blue. I could draw this forever, and this is the diagram that I gave you. But I can do this in a different way. I can eliminate the reproduction over here and simply look at the lineage through this end, and this is what will happen. And the reason why I do that is that I can reorganize this and present it in this way and finally this way. Because if we look at this way, Another way of thinking about bacterial reproduction is not that a mother produces two daughters, one old and one new. It is that the mother, which keeps the damage to herself, is really the old daughter. And bacteria are therefore a lineage of mothers, or the mother keeping damage to herself and budding off new daughters all the time. We think of yeast being this way, but we don't think of bacteria being this way. But the fact that the old daughter is keeping more of the damage, I would suggest another way of saying it is that the old daughter is really the mother, and the mother is keeping the damage to herself, and therefore budding off new daughters with less damage to give the offspring, the new daughter, a fresh start in life with a rejuvenated cell content that's free of damage. If this way of looking at things sits okay with you, the old daughter is the continuation of the mother keeps more damage to herself, and we can think then of the mother lineage as increasing in redness, accumulating more damage over time, and therefore aging. This is important because if that's the way of life, viewing life, we can think of eukaryotic multicellular organisms to be the same. A mother is born, she has little damage, but as she reproduces and makes offspring, that process is damaging. She accumulates damage and therefore picks up over life and eventually ages, and in this case, mortality ensues. We can ask then, are multicellular mothers also asymmetrical? If we think of the fact that multicellular organisms like us divide through a single cell zygote, a single cell zygote basically transfers very little damage to the offspring. This mother dog has cataracts, but she doesn't transfer the cataracts over to the daughter because the daughter gets a new cataract that's made fresh de novo from the single cell zygote. There are many arguments for why a zygote should be single cell, but this would be one more that we could add to the list. As a result, the adult that grows out of that single cell zygote has a fresh start in life. And because all the damage has been left with the mother, multicellular mothers are nearly 0 and 100% asymmetrical. A bacteria is different. I mentioned 40% and 60%. So a bacteria will simply represent a more primitive state, okay? Main difference between asymmetry partitioning between bacteria and eukaryotes is 40-60 versus 0-100. I throw in a question mark because this is speculation. The higher asymmetry rate in multicellulars could also be higher because multicellularity may require some different form of metabolism and cell growth that will give us a higher damage rate. If that's the case, that would have made multicellular organisms have mortality that's an obligate outcome of their lifespan. So this is a graph that I show you for E. coli, and you've seen this before. If we're to argue that multicellular organisms evolve by having higher damage rates and higher asymmetry, what that will do is basically elevate the red line so it does not intercept the line, right? Higher rate of damage and asymmetry. The other consequence of higher rates of damage and asymmetry is that by having the higher asymmetry, all daughters then will be born free of damage, or at least more similar to each other. So the blue line will become flat. The fact that we do not see a flat line with E. coli strongly argues that the mother is allowing some form of damage to slip in bacteria from the mother to the daughter. Therefore, the healthiness of the daughter depends on the healthiness of the mother, which is what this line represents. One would argue, therefore, that aging and asymmetry in bacteria is a more primitive form of what's going on in multicellular organisms. Given time, I think I'm going to skip through some of these predictions or statements and end by saying a few more specific comments. If you want an intuitive reason for why asymmetry is good, from the point of view of bacteria, people ask me all this, why is asymmetry good, okay? Why does a bacteria make a daughter that grows more slowly and one that grows more quickly? Asymmetry is advantageous from a bacteria for the same type of numerical theory as what is known as portfolio diversification in economics. If you have two bank accounts of $500 and you keep them both at 8%, they'll give, and that's having two daughters that are identical, that yields less money than if you have two bank accounts, one at 6%, less than 8, but the other one at 10%. The 10% account will grow more quickly through compounded interest and give the investment a higher yield in the long run. Lineages of bacteria, I would argue, are exploiting the same phenomenon. The new daughter, which has less damage, the newborn, the rejuvenated newborn, is like the investment at 10%, and aging is the account that gets less money, more damage, therefore 6%. Aging can result from wear and tear, okay? That's one way of aging, of course. The evolution of damage asymmetry accelerating the aging process beyond wear and tear. The final thought is this. Is this really then just bacteria, or can we extend it to other organisms? It is now appearing in the literature as I read it, because similar ideas are now appearing in discussion of aging multicellular organisms, and that is that people are contrasting lifespan with health span. Parents' shorter lifespan, aging, is the price that's paid to produce offspring with better health span. Bacteria mothers are aging because that's a price they pay to have healthy daughters. That could also be true with multicellular organisms. And this statement between lifespan and health span is a movement toward that direction. The reason why we age and die is because we're paying the price by shortening our lifespan to give us a better health span, higher health earlier in life, which is another way of saying we want to give our children a healthy start. Therefore, we're paying the price of aging and potentially death by giving our children that benefit. So I will end with this and stop sharing, and hopefully we can move on to questions. If there are questions, and we can still squeeze in with the time.
Okay, great talk. Thank you so much. There are questions, but Dan Weizmann's questions are too many. So maybe Dan, you can ask the question yourself, because I need to process them myself first. But Kim Reynolds is asking a question, and in the meantime, Dan, maybe you can unmute yourself and get ready to ask your question. Kim is asking whether there are any mutations in bacteria that tune alpha.
So we would love to find that, and we've been trying, not through screening of mutations, but we've been trying to select for bacterial lineages that are more or less symmetrical than what we find in the laboratory strains. We also scanned collections of E. coli that we had our hands on to see whether we found variation in the asymmetry alpha. Unfortunately, to date, we have not been able to find bacterial lineages that are vastly different from what we see in laboratory E. coli strains. In this case, it's the strain MG1655. We would love to find those because that will allow us to remeasure all these things. So we still have projects going on trying to elevate the asymmetry by selecting under very damaging environments, hydrogen peroxide, for example. But that has not given us an outcome. We have tried selecting for symmetrical bacteria. That also has not given us an outcome. So the value that bacteria seem to be um, seem to have evolved to holds very well for reasons that we don't truly understand.
Okay, thank you. Dan, do you want to go ahead?
Yeah, sure. So I have a sinking feeling that I've asked you this question before, but I forget the answer. So you show that you get less asymmetry when you have a higher damage rate. So that's kind of across experiments. And then I forget, do you see anything within a single experiment? Is there some kind of anti-correlation between amount of damage that the mother has and how asymmetric her daughters are?
I'm just trying to think whether we can do that. The problem with doing that is we have to use the whole population right now to estimate the parameters. And if we restrict it to one group of mothers with more damage and therefore longer doubling times, we lose fitting power. So if you saw, I mean, unfortunately the noise level is very high, right? So that type of finer analysis has been very difficult. So the movie that I showed you with the fluorescent reporter, that is going to be used with time to try to answer that question because it allows us to quantify the damage visually. Okay. So we didn't develop that system. The person that actually should acknowledge is Francois Tadet's group, who began this whole work. And they actually then worked on having reporters. So we have those, and if that data could be collected, it may give us a resolution to answer that question that you asked. Dan, you're frozen now.